Thank you. So, without any further delay, let me get into the talk. Yeah, I'm Senthil. I am head of Integrated Photonic Devices Group, as well as I'm deputy head of our department for education, looking after 120 PhD students and about 25 master students. So today I will describe about our university, our department, and also I'll specifically give you some research examples of what I do in my group. But that is not all about it. Our department and the university has wide-ranging research capabilities. Therefore, so I can't present everything what happens in the whole university. I will start with what I'm doing in my group. And then if you have any question, please do stop me while I'm presenting and also after the talk. Okay, so I'm coming from Southampton. I'm not sure how many of you know where Southampton is. Southampton is at the, okay, let me just open the laser pointer. So, yeah, so Southampton is actually at the south of England. So it's called Southampton. It's not very far from London, about 1 hour travel from London, similar distance from Heathrow Airport. And then we have about 6-7 campuses in Southampton. And also we have 2 overseas campuses, one in Malaysia, one in Delhi. And we were founded in 1862 as Artley Institute, but we have been a university for the last 75 years or so. And this is some of the metrics. So we are 17th in the UK, but depending on which ranking you see, we are normally top 100. And then we have about 140 countries from where the students to Southampton comes from. I was just telling this morning, Professor Ratno, that we are #1 in the UK for electrical and electronic engineering, where our department comes under. This is about our department, which is called Optoelectronic Research Center. We are the largest photonics institute in the UK. We were founded about 40 years ago, but yeah, we have been active in photonics or optics research from 1960s. We have wide-ranging topics. I will show you different groups in a minute, but as you can see, from starting from optical fibers through integrated photonics and biophotonics, which I will talk about today. And also we do nanotechnology, nanophotonics, and quantum technologies, which are related to your central research too. In addition to the research we do, our focus is mainly towards innovation, applied research. Therefore, we work and then a spin-off many companies. You will see we have about 18 spin-off from our department. I will show you in a minute. So these are some of the research groups in our school. Even though I call invariably department and school, we call ourselves school because school is bigger than department. But within our school, we have multiple research groups, as you can see, coming under fiber and communications, nanophotonics, and also photonic systems, circuits, and sensor, in which my group comes under, which is integrated photonic devices. We also have lasers and special fibers. So these are the actual research groups coming under 4 categories. So as I mentioned to you, we are the largest Photonic Research Institute in the UK and one of the largest in the world. And we do research starting from basic science to applied science and to devices research. As I mentioned, we are doing lasers and photonics research from 1960s. Many of the world leading innovation in optics and photonics happened in Southampton, starting from low loss optical fibers to erbium doped fiber amplifier, which was invented in ORC in Southampton in 1986, without which you cannot enjoy now the internet on your hand. So most of the internet infrastructure, which is responsible for the high-speed internet, had been linked with innovation in our department. And currently, the modulators, high-speed modulators, silicon photonics are also current topic in the ORC. Fibers invented in Southampton is already in Moon and Mars and in many space missions. We don't have undergraduate students. We only teach or train PhD students and master students, similar to BRIN institutes. We don't teach, but we mainly entertain research students. And many of our alumni are in the top roles in academia and industry because of the innovative research we do. We have a clean room facility, which is where most of our photonics devices fabrication is happening. And then we also have about 100 specialist research labs where we do the test and measurement of those devices prepared. And what you are seeing is the clean room layout. If you're interested, you may scan that QR code, which will take you through a virtual tour of the clean room where you can find out the facilities available. As I mentioned, we do applied research. Therefore, we collaborate with many industrial partners. As you can see, we collaborate with many industries. The big industries you can name, you will find them here. Either they are collaborating with us currently or collaborated with us in the past. As I mentioned, we have only 2 programs, master's and PhD. I will discuss more about the programs later on during the day, but just to give you an idea, we have master's in optical engineering, which covers most of the photonic materials, photonic systems, sensing, also AI topics in masters. The masters is normally in the UK 1 full year. So there will be 2 semester coursework, about 8 modules they study, and then summer project. So I will discuss with some of you more on this later on. My aim to visit Indonesia is to set up some joint masters, double degree masters. That's one of the aim. But irrespective of that, any of your students can just apply even without the double degree directly. We have also 3 scholarships up to £15,000. That's almost half of the tuition fee waiver given to a student who has merit. So we also have a PhD program, which is our flagship program, where you can, for example, Indonesian students come with LPDP support, or they can also apply for the scholarship available, which is even though limited for international students, there are scholarships available that they can apply for. Okay, so that's about the university, my department, very briefly. Now, let me just go into my own research group, and I will, for the next 30, 40 minutes or so, I will present some of the research activities that are being carried out in my group. But as I mentioned, there is wider, ORC or our department research, as well as in the university, there are many related research to your research center on nanotechnology or materials. So I will concentrate from now on the type of research we do in my group. So as the name says, integrated photonic devices group. Yeah. I work on integrated photonic devices, which are useful for applications like telecommunications, sensing, and also for on-chip or nanoscopy, super resolution microscopy. In other words, my recent interest is also on energy-related research, which is for photoelectrochemical water splitting, which I have started last year with 3 African countries funded by UKRI. So what you are seeing here are some of the examples of the integrated photonic devices that we have developed. What you are seeing here is the mid-infrared waveguides that is useful for mid-infrared sensing, as well as for developing mid-infrared spectrometer on chip. What you are seeing here is some metasurface for signal enhancement. And also what you are seeing here is the gas sensing platform. I will explain you in a minute. And then spectrometer on chip. And this is an on-chip platform for high-resolution microscopy or nanoscopy. And I will very briefly discuss about these chips. What you are seeing, these are spectroscopic chips for signal enhancement. So just very briefly, because I'm not going deep into this topic, as I mentioned, my recent interest is also on photoelectrochemical water splitting. This is one of the projects I've just started with a few African countries. The aim here is to develop most efficient photoanodes for utilizing solar energy to directly convert water into hydrogen and oxygen. And the hydrogen produced could be used to directly fuel, for example, vehicle, or convert hydrogen using fuel cell to electricity. So this is the aim, and what I'm currently doing is making use of materials that are abundant in nature, for example, hematite. So heterostructuring hematite with 2D materials. Therefore, we can improve the efficiency of hematite, which is currently less than 10%, solar to hydrogen conversion, whereas our aim is to exceed that current limit of 10%. So that's something happening, but I will not present so much more on this. But if any of you have got interest, we can always collaborate on this. Okay, so the rest of the talk I have split it into 2 parts. One about the integrated photonic devices, and 2 on the applications, which is biomedical applications. So initially, let me present you on some of the devices we have made in the last few years. The problem is that I went to this university 22 years ago. So there are a lot of things happened. So I cannot present everything I have developed. So what I'm going to present is just some snapshot of few things I have done. But then the second part, the biomedical spectroscopy or the applications, is my current interest. So that's how I have arranged this. But this is not all we do. This is only, as I mentioned, a snapshot of a few things we do. Okay, so in this first part, let me just present you about some of the waveguide based devices and then some nonlinear waveguides and on-chip spectrometer. So just to show you some of our interest in waveguide based devices, as you can see here, we developed many different platforms, including erbium, neodymium, thulium, ytterbium, doped waveguides. Because you are materials researcher, you know, active materials or rare earths are the basis for the energy transition, and therefore they are useful for lasers or amplifiers. Therefore, we produced many waveguide lasers, including single mode lasers, you know, producing a monochromatic wavelength. But these are not big lasers, these are small lasers on chip. Something like a centimeter square, you have a laser pumped by optical means or using laser diodes, for example. And also we have developed many waveguide amplifiers. For example, erbium doped fiber amplifier is famous, but then they are in fiber form. But if you want to do some signal processing, you know, in the lab or at the you know, exit point of the communication end, then you need amplifiers on chip. So we developed erbium doped waveguide amplifiers also in other rare earth material, and then we also worked on chip resonators. These are useful for you know, filters, for example, optical filters, and many different functions on chip using resonators. Just to give you some more interesting stuff about resonators, what you are seeing is a microsphere sitting on top of a waveguide, and that was the research I did, I think, more than 10 years back, where we produced integrated microsphere laser, that is a microsphere doped with rare earths sitting on top of a waveguide, and that produces lacing. So what you are seeing is the lacing from the microsphere, but pumped using waveguide and the collection using waveguide. So many of you know that high resolution microscopy is required if someone has to image a cell, for example, or an organelle, where we need resolution better than what a normal microscope can offer. A normal optical microscope can produce only maximum resolution of 200 nanometer. That is because of the diffraction limit imposed by the visible light we use. So what we have developed here is an on-chip spectroscopic platform where the beam shaping happens on the chip, which is nothing but the structured illumination microscopy or fluorescent microscopy that happens on the chip. The traditional chip used was silicon chip, where people have demonstrated super resolution microscopy. But the problem is that silicon is opaque in visible wavelength. Therefore, if you want to use conventional microscope available in a biochemistry lab or a biological lab, which uses inverted setup, where you have a cover slip on which you have the sample and the light has to go through the cover slip. Whereas when we use silicon, we cannot do that. Therefore, what I have developed is a transparent on-chip nanoscopy platform where we can use regular microscopes available in a hospital or in a biochemistry lab. So what you are seeing is a cell here, where we are showing the improvement we got with the transparent platform I developed, where we are seeing a few tens of nanometers of details with the inverted microscope using the transparent waveguides we developed. So similarly, we have also developed gas sensing platform. For example, what you are seeing is a waveguide here, which is suspended, and therefore you have the gas which can interact all 360 degrees with the waveguide here. And what we have shown here is the enhancement of the waveguide platform offering compared to a free space light, 108% efficiency, and for a free space gas sensing, you have to have few meter length of gas cell, or in a multiple reflection configuration, maybe few tens of centimeter. Whereas we have showed better performance within a centimeter square chip. And this was used for acetylene gas sensing, but we are also currently doing methane gas sensing using similar platform. Okay, now I will quickly show you some of our aspirations towards waveguides useful for spectrometer on chip. So I will present this in two different ways. One using electro-optic platform, the other one using thermo-optic platform. I'm not sure how many of you know the concept of spectrometer. For example, if you take any regular spectrometer, for example, FTIR, there are two arms, and one of the arm is moving where you change the path length. Therefore, when the two beams interfere, they produces interference fringes, which is then Fourier transformed to get the spectrum. So basically, you need to have two arms of the light. For example, like this, what I'm showing here is a Mach-Zehnder interferometer, but in regular free space spectrometer, you use Michelson interferometer. But basically, these are interferometer. So one of the arm path length should be changed because these are fixed arms rather than a Michelson interferometer where the mirror moves. So what we can do is we can change the refractive index so that the path length can be changed. That is the idea here. And the refractive index change can happen in two ways. One, using electro-optic effect by applying electric field. Then you change the refractive index of the material. Therefore, the path length on one side is changed. The other way is to introduce heat. So that is using the thermo-optic effect where the refractive index is changed as a function of temperature. So these are the two principles we used and developed two different platforms. The first platform is based on induced Chi2 nonlinearity, which is nothing but the electro-optic effect. The second platform is based on the chalcogenide waveguides, which when applied with temperature, you change the refractive index. Let me quickly show you some details about the electro-optic platform, where the best material for electro-optic activity is lithium niobate. But if you are working on lithium niobate, you know that it's very hard to produce integrated devices based on lithium niobate because of the hardness and the directionality and all the associated property of a crystal. So there is an alternative available to produce high refractive index contrast, which is lithium niobate on insulator, but it's very expensive and it introduces a lot of steps. Therefore, what we have come up with is an amorphous material platform, which is nothing but a thin film you deposit on top of any substrate, and then you apply what is called thermal polling to induce second order optical nonlinearity. Because amorphous materials or glasses don't have Chi2, you may know, because of the isotropic nature, but we introduced the Chi2 by breaking the symmetry, and we achieved Chi2 nonlinearity up to about 29 picometer per volt, which is very close to what you can achieve in one of the directions of lithium niobate, which is 35 picometer per volt. So we applied this, and we have also shown that after getting the Chi2 established, after the waveguide has been fabricated, the Chi2 is still available without any decay. So what we are showing here is the sustained induced Chi2 in this material, which retained after the fabrication of waveguides. That means now our waveguide platform is available with Chi2 property, and therefore we can fabricate electro-optic waveguide. And here I'm showing some of the simulation we have performed, and these are the electro-optic waveguide, which are poled. Then they have gold electrodes, for example, here to apply electric field to change the refractive index. Therefore, the work is still ongoing, and this will form a spectrometer on chip. Similarly, chalcogenide waveguides, if some of you are interested in chalcogenide, you know that they are very delicate material to deal with. Therefore, we found a good way of fabricating waveguides which are like this. So you have silicon pre-patterned, then we deposit chalcogenide layers. For example, Ig2, what I'm showing is one of the chalcogenides, which is germanium arsenic selenide, and then Ig3 is germanium arsenic selenium and telluride, and Ig3 has got higher refractive index. Therefore, we found a waveguide cladded between low refractive index, and we formed same Mach-Zehnder interferometer on chip, and these are the spiral to increase the optical path length. Therefore, the resolution of the spectrometer is better. What you are seeing is the loss of the waveguide. We achieved 0.1 dB per centimeter, which is the lowest achievable so far in chalcogenide. And we have calculated thermo-optic coefficient using that setup I showed in the last slide. And we estimated a thermo-optic coefficient of 1.1 into 10 to the minus per Kelvin, which is similar to what you get it in silicon, that is 2 times 10 to the minus 4. So then we have fabricated spectrometer on chip, and the characterization of this is still ongoing. So those are some of the on-chip or integrated photonic devices. Now I will switch to some applications of these devices, in specifically in two biomedical applications, one in respiratory, the other one in cancer. So the idea is like this. I'm sure you know, when you have some problem, you go to doctor. Doctor is not God. They have to still do diagnosis to find out what the problem you have. So they take blood, but then if the condition is very serious or they don't know what the condition is, they have to send it to a centralized lab to do the analysis. But the result is not immediate, right? Then you come back home, the next day the result comes, and then you go and then get treatment. But if the condition is very serious, then one day or few hours is a long time. So what we are developing in my lab is to handle this situation. So instead of going through the centralized lab to get the result, we would like to use the chips, optical chips we fabricate in our lab, we develop in our lab, and use them to analyze any body fluids or even tissue, and then use them in the clinic with a compact spectrometer, and then produce the result within, say, 10 minutes or so that the doctor in the clinic will tell you, okay, you have this problem. You have to get this treatment or you will be treated immediately. Okay. So that is the ambition for which we are going to use optics and optical chips. Okay. That's what my final talk is going to be, part of the talk. I'm not sure how many of you know spectroscopy. That's why this is, because I prepared for talk among a high school student. I thought maybe I'll just insert that one slide here to explain if you don't know spectroscopy. But I think you all know, but still, spectroscopy is nothing but scanning a wavelength and finding where the absorption is. So for example, the leaves look green because they absorb, or the chlorophyll within the leaf is absorbing all the wavelength in the solar spectrum or white light, except green. Green is reflected. That's why the leaves look green. Okay, so that's the concept we are going to use, which is spectroscopy, where if any of our biological or biochemicals, which are nothing but the biomarkers, are absorbing a specific wavelength, then we know what that chemical or what that molecule is. So this is the principle we are going to use. So by using this principle, spectroscopy, which we call it as vibration spectroscopy, because molecules are in constant vibration, okay? And they vibrate along a bond between the two atoms. For example, if you consider H2O, the two hydrogen and one oxygen link with a bond, and these bonds are constantly vibrating, and that vibration has got the energy, and that energy is provided by the light which falls on them. Okay. For example, well, it doesn't need to be energy, electromagnetic radiation. That's why when you put food in the microwave frequency is absorbed by water, and therefore it vibrates, and this vibration and the friction between these different molecules with the food heats up. Okay, so this is the concept we use, and there are two types of vibration spectroscopy. One is IR spectroscopy, the other one is Raman spectroscopy. One is absorption in absolute terms, the other one is absorbed light plus whatever comes out, that is the shift. So when it is Raman, it's the Raman shift. When it is IR, it's the absolute wavelength absorbed. Whatever you use, then many molecules in our body, which are lipids, nothing but fat and other lipids, proteins, DNA, RNA, carbohydrate, all of them are biomolecules. Therefore, they have unique absorption, which are called fingerprint absorption. So based on that, we will now use, using this concept, we will do many different biomedical sensing or diagnostics. So there are many different types of disease diagnosis proposed and demonstrated using vibration spectroscopy. But what is missing so far is what I mentioned. There is no specific device that is capable to do diagnosis at the bedside or at the clinic. Spectroscopy so far happens only in the lab. So my aim is to simplify this and make it possible at the clinic. Therefore, there are two things we do. One, well, two things different to what has been done in the literature. One, miniaturized on-chip spectroscopy, so that you need very little sample. What we use normally is between 4 and 10 microliters. Yeah, 10 microliters maximum. And the second one is quantification. So far, people say, yes, you have this or not. But what we wanted to do is quantify, okay, how much of the problem you have. So this is what will be different. Normally, IR spectroscopy is done using what is called ATR crystals. ATR is nothing but attenuated total reflectance crystal. So I have a crystal, I put a drop of blood, then light goes through the crystal, get bounced up at the top interface where the sample is, and then come back. Where the bouncing happens here, like what I'm showing, there's an evanescent field, and that evanescent field interacts with the sample and produces the absorption spectrum. So that's the technique used. But these crystals, you can see, they are about 10 centimeter in length. So they need more sample volume, which you cannot find in one of the application I will tell you in a minute, babies, respiratory distress syndrome, where the babies have got tiny sample in their lung. So you cannot hold 10 centimeter long crystal. Therefore, that's not possible. Also, in a clinical environment, you cannot keep cleaning the big crystal because there's a cross-contamination. You don't know whether the problem is with the current patient or the patient you diagnosed earlier. So what we are developing is these chips based on silicon, and they are very cheap, something like $1 per chip. Therefore, you can just dispose it off. So you don't need to use it, clean it, and then use it again. So these are, one-time use. And also the top side you are seeing, the colorful one is because they are structured. They are metasurface. They increase the signal. Therefore, it's giving you two purposes. One, to increase the signal, like this metasurface. You can see without microstructuring, you get very low signal. Then with microstructuring, we get very high signal. And also they are cheap, so you can dispose it out. So these are the advantages. And again, I'm just emphasizing what the approach we propose is just put the sample, a blood drop on the chip, put it in a compact spectrometer, measure the spectrum, and we do lots of AI and we quantify the amount of biomarkers. So this is the approach we do. Quickly, I will show you some of the application. So today I'm going to talk about two applications. One is neonatal respiratory distress syndrome. The second one is cancer. So let's talk about the first one, neonatal respiratory distress syndrome. I am sure many of you, if you have gone through the phase of having baby, you know that the gestational time is, say 35 weeks. So if a baby is born prematurely, say at 30 weeks or 28 weeks, they are too tiny and their lung is not developed well. So this condition is called respiratory distress syndrome if the prematurely born baby's lung is immature. They are not fully developed, then they have problem breathing. So now, how the breathing happens within the lung? We have something called a surfactant within the lung lining. The surfactants reduce the surface tension of the pores, which is called alveoli. So these pores open and close, and then let the exchange of carbon dioxide and oxygen between, the lung. You take it in and then supply it to blood, and from the CO2, which is unwanted, comes into the lungs and come out. So this is keep happening, but this is with the help of what is called a surfactant. It's called lung surfactant. These are lipids. So currently, there is no diagnostic test to find out in the clinic whether the baby has got enough surfactant or whether the lung is matured or not. There's a treatment for it by giving exogenous or artificial surfactant to the baby's lung to enable this function. But there is no diagnostics. So there is a treatment, but whether they are really having that problem, there is no diagnostic. So what the clinicians do is they just sense, okay, this baby is struggling to breathe. Let's give artificial surfactant. But if this breathing problem is due to some other cause, then they are making it worse. They are not treating the condition, rather they are complicating the situation. Therefore, what we are proposing is a technique where the lipids, the surfactants, there are two lipids. One is called DPPC or lecithin. The other one is called sphingomyelin. The ratio of these two lipids decide the maturity of the lung or the presence of enough surfactant. So a ratio of this L to S at 2.2 or more means the lung is healthy, they have enough surfactant. If it is LS ratio is below 2.2, that means it is a diagnostic for NRDS, that is neonatal respiratory distress syndrome. That means the baby has to be treated. So what we have developed is a spectroscopic technique. What you are seeing here, you see, there are two spectra. You see one for lecithin, one for sphingomyelin. They have very distinct peaks here. You see, absorption peaks. So based on this, we can now develop spectroscopic approach to quantify the actual amount of lecithin and sphingomyelin. Therefore, okay, we developed a complex study, and then finally, what you are seeing here is the lipids, the sphingomyelin, lecithin, and then we used three different sample. One is bronchoalveolar lavage, that is, sample deep from the lung, and then tracheal aspirate is, the pipe connecting between the lung and the throat, and from there, and also sputum. So what we have studied, and it showed that the sphingomyelin and DPPC, these are synthetic, we measured, is clearly identifying those DPPC and sphingomyelin in the actual sample collected from the human also. And then we have used, we have done about 3,000 spectra. As I mentioned, we used, less than 100 microliters of sample and dried. And as you can see from our spectroscopy using machine learning, we have then found the LS ratio. The LS ratio here is more than 2.2 because this is from a healthy volunteer. Okay. So that is the application in respiratory distress syndrome. Let's now go to cancer. When it comes to cancer, it's unfortunate, no one should have cancer, but so many millions of people every year develop cancer. So the routine diagnostic pathway is that when they go to the clinic, they will have to go through tissue biopsy. It's either like a pinch biopsy or they cut a part of the tissue, send it for histopathology examination in the lab, and then the result comes later. But then when this is at the very early stage, that's not going to be detected. And also the other modality used is CT scan, CT imaging, but then none of them are providing molecular information, what is really gone wrong. So what we are developing here is a liquid biopsy approach where using blood or any body fluid, how to detect cancer, especially how to quantify the cancer. So I'm going to show you three different things. One, something about the DNA, then something about the protein expression, and finally about epigenetic modification, which is nothing but the change in the methylation state. Okay, so we are going to use ATR, FTIR, and the Raman spectroscopy. So in general, vibration spectroscopy to do this. So let's come to this, right. I'm sure we have DNA in our body, right? The DNA is present inside a cell. The DNA inside the cell is very long. If I consider one unit as one base pair, that's how it's called in cancer biology. So one unit of this molecule in DNA is called one base pair. And the cell, the DNA in the cell has got so many millions or few billions of base pair length of DNA. So the DNA inside the cell is so long. But then you also know that continuously in our body, cells are dying and new cells are developing. When the cells are dying, when the cell death happens, the DNA inside the cell get fragmented. These billions of base pairs become fragmented into hundreds of base pairs. And normally, these are, when the fragmentation happens, it shed into the blood. So the cell that happens, the DNA get into the blood, and in the blood, if you see, the DNAs are about 167 base pair in length. Okay, that's for a normal human being. A normal person's cell is dying, and the DNA is released into the blood, and these are called cell-free DNA. Okay, cfDNA in the blood has got a fragment length of 167 base pair. Just 10 years back, they found that, similar to cell-free DNA from a normal human being, when someone is having a cancer, from the cancer cell, and when the cancer cell dies, also the DNA from the cancer cell is released into the bloodstream. And these are called circulating tumor DNA, because these are coming from the tumor cell. Therefore, those DNAs in the blood are called ctDNA, circulating tumor DNA. This ctDNA is shorter than 167 base pair length. Don't ask me why, because I'm not a cancer biologist. But also, I don't think it's clear yet, even to the cancer biologist, why the fragment length of cancer DNA fragments are lower than 167 base pair. Okay. For us, what we need to remember is the normal cell produces 167 base pair fragments. The tumor cells produces much less, say 150 base pair length of DNA. Okay. So currently, this is not available in every clinic, but it is still available in lab-based setting. For example, when someone is diagnosed with cancer using tissue biopsy or CT scan, then to confirm and to specifically find out what type of mutation, what type of cancer they have, then people do what is called sequencing, DNA sequencing, to find out all these changes in their fragment length. But it's very expensive, something like 5,000 pounds or maybe $6,000 per measurement, and it takes so many days to do the sequencing. So what we have developed is a method to measure, not identify, to quantify what is the length of these fragments in the blood. Okay, so we used ATR, FTIR, and Raman spectroscopy to find out the difference. So initially, we do machine learning. Therefore, we have to train the machine learning model. Therefore, we start with known fragment length first, and then we train the model. Then what we find is, initially an R square, which is the how good the model is working, find 94% is good. And then with the root mean square error of 22 base pair, which is still not good. So we carry on developing this model, with many different complex mixtures containing different fragment length. And we developed an approach here, which finally used 11 biological samples. These are DNA fragments produced from rat DNA. And we used 6 of the samples to train the model, and 5 you used to test. And what you are seeing here is the distribution of the fragment length in the blood, the blood from where the DNAs were derived. And what you are seeing is the distribution. So based on the median length here, you can say whether there is cancer or not. Okay, so this is the approach we developed, and which is going to be published soon. We submitted the revised manuscript. So just to test, so that's the approach we developed. Just to test, we have now taken two different tissue samples here. One is a healthy part of the liver. The other one is the cancer part of the liver. I'm sorry, yeah, it looks a bit irritating for you, but that's how we look inside. So I have a student who is a biochemist working with a cancer surgeon who's my student's co-supervisor. So we get samples when they operate. So this is the real human sample we got from University Hospital Southampton, where I have collaboration. So what you are seeing here is, there are three cancerous sample, one non-cancerous sample, right? So we do see differences here. You see, in the spectroscopy we have done on these samples. So we used these. What you are seeing is, okay, these are 5 cancer healthy sample, 5 cancer sample. You can see the differences here. You see in this, but maybe as a human we cannot exactly identify what the actual change, etc. But the machine learning is very smart. It understands, and then what we have done here is the classification, just classifying which one is cancer, which one is non-cancer. But that's what the machine learning has done. But that's not what exactly we wanted. We wanted to quantify, right? So what you are seeing here is a cancer-derived fluid. So we take this tissue and then take out what is called tissue interstitial fluid. The fluid which comes out of the tissue, which brings in all this DNA information. So we used that, and then what you are seeing is the cancer. You can see the median length is above 167 base pair, and then below 167 base pair, whereas the non-cancerous one is above or the healthy one. So just to give you, so this is the gold standard, which is the electrophoresis, which normally people do to find out the true length. And we found that the gold standard and our spectroscopy is exactly matching. So now what you are seeing here is the actual result. You see, the cancer fragment length is much lower compared to the healthy fragment length, correct? So this is the whole idea of the study, and we found that spectroscopy is really doing that. Okay. So there are many ways you can find out cancer. One is this fragment length. Then when cancer happens, the methylation state of DNA changes. Normally there is unmethylated DNA, but there is always some form of methylation in our DNA, but the methylation percentage changes. And we found that spectroscopy can also estimate the percentage of methylation. And this is what we show here. For example, what you are seeing is unmethylated, methylated, and hydroxymethylated. These 3 forms are producing distinct features in the spectra, which we use. And what you are seeing is the classification first before going for quantification. So our spectroscopy very clearly classify which ones are unmethylated, which ones are methylated, which ones are hydroxymethylated, because these percentage changes when someone is developing cancer. So based on that, we can predict whether someone has got cancer or not. So further we trained the model. What you are seeing here is a ctDNA, circulating tumor DNA, where the methylation change is happening. Okay, here what we have done is we have artificially varied the methylation percentage in a ctDNA, and measured, how accurately, we can quantify. So what you are seeing is the actual measured value compared to the true value. And you will see that they are not too different here. So that's one thing. Then finally, I'm sure you may know that the cancer is also affecting women more than men in many cases. For example, ovarian and breast cancer are some of the deadly ones in women. So we work very much on this. In fact, I just got this year a new project with Africa to educate them on the ovarian cancer and endometriosis in how to deal with that, as well as to diagnose this using spectroscopy. So for this cancer, especially ovarian cancer, has got links with what is called a cancer antigen 125 or carbohydrate antigen 125. The amount of this protein is a protein. This protein is expressed more when a woman has got ovarian cancer. So the normal percentage is, normal amount is 35 units per ml. When someone has got ovarian cancer, this amount is increasing. So by measuring the amount of CA125, which is the biomarker, then you can say someone has got ovarian cancer or not. So it's not only CA125 ovarian cancer, but we also measured various other protein biomarkers responsible for various conditions, various cancer conditions, and we showed that we can discriminate and classify between these various protein markers. And finally, what we have shown is, okay, what you are seeing here is the CA125. We varied the amount. For example, I told you 35 units per ml is the normal physiological concentration of a normal human being. When they develop cancer, the amount will increase. And what you are seeing here is when the concentration is varying, how the spectra is different. But again, machine learning produces better understanding than us. So what you are seeing here is the preliminary result of measuring CA125 in human serum, that is blood. So currently, we have accuracy to the level of 8 units per ml. So if someone should have only 35, if they have 100, I think plus or minus 8 units per ml is still a significant measure. So these are some of the research we do. Just to summarize, I think I convinced you that the integrated optical devices and the spectroscopy approach we developed is useful for biomedical diagnostics, and the main aim is to move the diagnosis closer to the bedside, and this will happen hopefully because of the miniaturized and most efficient devices we are developing. So thank you very much. These are some of the people who are responsible for or contributed to this work. This is my current group members, postdocs and students. Maybe some of them are still missing, and these are very few of my collaborators. I can't put everyone here, and I thank the funding I got from these people. Thank you.